Okay, hello and welcome back to Genomics in the Virtual Lab. And I wanted to start by giving a brief overview of what we did last week. I know that maybe not everybody's joined today just yet, but hopefully everyone will join. We are going to record this session, so it's going to be available later. And after we review what we covered in last week, maybe we can talk a little bit more about your project proposals. I know that there are a few. So we'll take a look at those. And then we'll talk about how to develop a research project. So we'll talk about the different steps that are involved and a little bit more clarification about how it is done in the context of bioinformatics. So last week, we talked about how protein structure and function are related and how they are studied by studying the relationship between sequence variation and structure variation. We talked about how the different types of sequences that are available at the nucleotide or at the protein level are, it is possible to study them using multiple sequence alignment. And also we can see the variation at the functional level and different methods that we have to study that variation. Now, at the end of the first week, the idea was to get everyone to be able to be familiar with some of these tools, try them a little bit hands-on, and then to be able to develop a proposal or a presentation about some topic related to what we studied. And I think a few examples were given where you could be potentially starting from. So in this week, we will continue speaking about the same topics, but we will talk a little bit more about how genomics is used to study virology and infectious diseases, how we can study the microbiome, and how antiviral drugs, vaccines, and antibiotics are developed and studied in the context of their effectiveness. So we'll hopefully take a lot from what you've learned in the first week and kind of apply it to a new set of challenges. And we'll talk a little bit about the technologies that are enabling us to be able to answer those questions. So last week, Sona Vasudevan and her collaborators were able to share a lot with you. And I think that the majority of you did really well. I saw a lot of engagement and a lot of interaction. So hopefully this week will continue in the same way. But obviously, every time that you kind of change the way things are delivered and the way that they are discussed, you know, it's a little bit different. So we're going to have to adjust a little bit and get to know each other again, kind of get into the same mode of learning. And, you know, some of the things that Sona was able to teach you are, of course, very, you know, good and detailed. And, you know, hopefully, instead of doing the same, we will kind of build on that and discover some new things in the context of this program. Okay, so in the weekend activity, you were asked to prepare 3 slides that introduce the different topic, a topic of your choice. And explain some of those using some visual tools. So let's take a look. I have here a list of those that were able to submit their weekend activity. Does anyone want to present their video or present their slides and speak a little bit about their projects? Are there any volunteers? So I see the first one here is Megan. Megan, would you like to share a little bit about your project, or would you like us to just take a look at your video? Yeah, you can continue to submit. In fact, we'll continue to talk about these projects. So yeah, go ahead, Megan. You can unmute yourself and even share your screen if you would like to.
Okay. Awesome. So Let me pull up my video quick. So I did record myself, so it'll just be like me talking on it. Okay. Okay. All right. Here's this.
Okay, so actually, I don't think we can hear you very well when you are playing the video.
Oh, really?
Do you want to just talk about the slides? I mean, you probably have the slides, right?
Yeah, totally. Okay. Let me exit out of this. Okay, I'll just pull up the PowerPoint then. Okay, so I chose phenylketonuria as my disease, and it's like it's an autosomal recessive disorder, so it comes from both parents on the gene PAH, and it causes a deficiency in phenylalanine 4-hydroxylase. So when this enzyme isn't working, that means the phenylalanine in your body isn't being converted to tyrosine. So the buildup can result in like brain damage. So, and I got most of that information from Uniprot. The accession number is just on the side here. So, and then here's just a picture of that, like what's happening. So the phenylalanine hydroxylase, that's not happening in the body. So it's just building up phenylalanine and it's not being converted. It's located on chromosome 12. I found that on OMIM, specifically this number. So I guess that's just like the specific location that it told me. Some more diseases associated with it. So phenylketonuria is when the phenylalanine levels are above 1200 mmol, and it said that the normal is around 100 mmol. So just to give you reference, that's like very high in the body. So again, like it's when hydroxylase is not happening. And then another one was non-phenylketonuria hyperphenylalanemia. That's a super mouthful. But that's when they're below 600 mmol. And then hyperphenylalanemia is the mildest form of it. So here's one like angle of the structure I found on the protein database. And I used the ID 1PAH. I wasn't sure like specifically what it was because I know with sickle cell or anemia we use the HBS versus HHB. So yeah, that's what I came up with. And then the PFAM domains were in ACT and biopterin. So those are just like the areas that it's found on the whole length of it, which is 452 amino acids. And then here's another angle of the structure, the ligand interaction. So I just zoomed in on the center and highlighted it in that lime green. According to SCOP, its class is an alpha and beta protein, so A plus B. And the fold is ferredoxin-like, superfamily is ACT-like, family is phenylalanine metabolism regulatory domain, and the domain is the specific phenylalanine 4-hydroxylase. On PyMol, I did a fetch on the PAH, and so you can see like the helices and the loops, which I straightened out, and then you can see like the beta strands. So here's another angle. I wasn't sure specifically where the like the disease-causing mutation was, so I fetched PAH and 1PH, and this was the difference in the corner. So I'm not sure if that's exactly correct, but that is what I tried to. So, and then here's the electro image of that. And then yeah, all my sources were just from OMIM, the PDA or PDB, and Uniprot. So yeah.
All right, great, thank you. Yeah, I think that's a really nice presentation. So it's a really good place to get started, right? So you're kind of familiarizing yourself where that gene is, where that mutation is, what the protein structure looks like. And there are obviously more questions, right, that you are probably left with after you've done this little study. So I think that's a good start. Let's take a look, and thanks for sharing that. Betsy, Betsy, do you want to share yours? Betsy is on here. Okay, maybe Betsy is not here. Let's see, Soprano Das. Soprano, would you like to share yours? Okay. And Kylie Eckert. Kylie, would you like to share yours? Yeah, so Soprano, I saw your video recording, and that's good to have. But if you want to just go through your slides and kind of explain where you are with that, or do you not want to do that? Okay, yeah, Kylie, if you want to do that, you can unmute yourself and share your screen. Okay, Soprano, that's not a problem. Or Soprano, sorry. Okay, no problem. So Kylie, can you unmute yourself? Yeah.
Okay, hi. Can you hear me?
Yep, we can.
Okay, I have headphones in and sometimes that messes up the audio. Okay, sharing my screen. Can you all see my screen?
Yep, we can.
Okay. So I picked a disease that was not one of the listed ones because it's something I'm personally very interested in. Unfortunately, there was not a lot of information about it, so that was a little more difficult than I anticipated. But I was researching autosomal dominant polycystic kidney disease, and it is caused by a mutation in the PKD1 or PKD2 gene. And there's a lot of mutations that have pathological significance, but there's not like one mutation that causes it. Polycystic kidney disease causes cysts to form in the 5th decade of life, and it is passed on from your parents. If your parents have it, then you have a chance of having it. The Uniprot accession number was P98161, and it was reviewed. It had a 5 out of 5 annotation score, and there was experimental evidence. It was a bit confusing because on Uniprot, it showed both polycystin 1, which is the protein produced, and PKD1, which is, I'm pretty sure, also a protein. And it was really confusing because sometimes there would be a model of one and sometimes there would be a model of another. So I tried to include models of both because it seems that mutations in both cause issues. It's located on the 16th chromosome, as you can see from this diagram here. I don't know if you can see my mouse, but, and the only disease that it's involved in is polycystic kidney disease. Whoa. This number, the 1B4R is the PDB ID for polycystin 1, which is the protein produced by it. And I will show you models in a moment. And I wasn't 100% sure what the domains meant, so I included what I found on PFAM. Then I blasted the sequence with a bunch of other non-human PKD genes and stuff because there were no other reviewed sequences. The human PKD1 gene was the only reviewed sequence. And now we have some models that I took from PDB. This is both PKD1 and PKD2. And then this is polycystin 1. And there were no ligand interactions for either. So then this was a diagram I found on SCOP with the family, superfamily, fold, and class. And I'm pretty sure this is for polycystin 1, this one right here. In PyMol, I used fetch and I used this one whose PDB ID was 6A70 because when I tried to fetch polycystin 1, nothing happened and it didn't come up. So clearly the mutation is in 6A70, which is this complex, which is PKD1, PKD2. And the mutation, one of the mutations that I found this mutation by on Uniprot, scrolling through all the pathological mutations under polycystic kidney disease and looking through the possible mutations in PyMol until I found one that matched up with the ones in Uniprot that would have pathological significance. And the first one I found was at position 3,285 and is a change from a V to an I. I'm not going to even try to pronounce that. And this is the whole molecule up here, and the mutation is over here, as you can see in this more zoomed in image. And then finally down here. I couldn't find an electrostatic model for the complex PKD1, PKD2, but this is an electrostatic model of 1B4R, which is polycystin 1, the protein produced. And these are all the works I cited in this presentation. There were a lot because it was really hard to find information.
Okay, so a really good, number of slides. You have 11 slides, so that's a lot. And but the good thing is that we can obviously see that you have done your research and kind of found what you could. Obviously, more questions are gonna still be there, and hopefully we'll talk through some of the process today. So maybe as throughout the week, you'll have more time to refine that presentation. Okay, great, thank you. And okay, so the only other one that I have, Suparno, Megan, I guess, are not going to be sharing today, but for the rest, or Megan is, sorry, you did share, right? Okay, Suparno, do you want to share now? Okay, Betsy is not here, sorry. Okay, well, Suparno, let's take a look at what you have. Can you, okay. I think you need to
Hello everyone, my name is Shoparno Dash and today I am going to deliver a presentation titled Phenylketonuria, a Structural Insight. So classical phenylketonuria is an autosomal recessive disorder which is caused by mutations in both alleles for the gene for the enzyme phenylalanine hydroxylase or PA. This is its enzyme commission number. This gene is found on chromosome 12 of humans. The phenylalanine hydroxylase enzyme catalyzes the conversion of phenylalanine to tyrosine in the blood with the help of a ferrous ion, oxygen, and a tetrahydrobiopterin molecule, all of which are bound to its active site. Now, certain mutations in the gene for phenylalanine hydroxylase might hinder the formation of the quaternary structure of the enzyme, which can lead to the increase in the blood concentrations of phenylalanine. The normal concentration of phenylalanine in the blood is 100 micromoles. When it goes beyond 1200 micromoles, then that condition can be characterized as phenylketonuria. Unless a low phenylalanine diet is introduced early in life, the victim can suffer from mental retardation. There can also be other symptoms such as light pigmentation, rashes, epilepsy, hyperactivity, psychotic states, and an unpleasant mousy odor. One of the mutations that can lead to phenylketonuria is a missense mutation which changes the codon from GAC to AAC. Since GAC codes for aspartic acid and AAC codes for asparagine, thereby residue 415 of the protein molecule is changed from aspartic acid to asparagine. In the DBSNP database, this can be found under the entry RS62644499. The PA gene can be found in the UniProt database under the accession number P00439 and the entry name PH4H_human. There is a gold star beside its name, which means that this entry has been curated and reviewed by SwissProt. Therefore, it is extremely likely that this information is reliable. It also has an annotation score of 5. In the OMIM database, the PA gene is registered under the ID 612349. It causes a total of 3 diseases: phenylketonuria or PKU, non-phenylketonuria hyperphenylalaninemia or non-PKU HPA, and hyperphenylalaninemia HPA. All of these diseases are caused by various mutations that affect the PA gene, which can be missense mutations, deletion mutations, and various others. Now, there are various protein structures available for the phenylalanine hydroxylase enzyme in the protein data bank, 2 of which I am going to show here. The first is the tetrameric human phenylalanine hydroxylase, which has the PDB ID of 2PAH. The second one is the catalytic domain of the human phenylalanine hydroxylase enzyme in complex with tetrahydrobiopterin, ferrous ion, and thioenylalanine, which is a phenylalanine analog. It has the PDB ID of 1KW0. Now, in the PFAM database, the chains A and B of the phenylalanine hydroxylase protein fall under 2 different domains. The first one is the biopterin-dependent aromatic amino acid hydroxylase or biopterin H with the accession number PF00351. Residue 119 to 449 fall under this domain. The second is the ACT domain with the accession number PF01842. This domain contains the residues 35 to 100. When I ran a blast against the sequence of the PA gene in UniProt, there were 3 verified homologs which showed up. All of these were PA genes in different other animals like bovine or cattle, mouse, and rat. Here is a tree of the 3 verified homologs, and these are their entry numbers. The phenylalanine hydroxylase enzyme exists as a tetramer in its normal biological assembly. This is a structure which has been provided from the PDB. This is a model of the active site of the PA enzyme while it is bound to a tetrahydrobiopterin molecule, a ferrous ion, and a thianenylalanine molecule, which is a phenylalanine analog. Here we can see that the geometry of the ferrous ion is an extremely distorted square planar, and also the thianenylalanine molecule is interacting with pi stacking interactions with the imidazole ring of the histidine below it. Now, the binding of the tetrahydrobiopterin molecule to the active site of the enzyme induces a conformational change through the entire molecule, causing the whole molecule to adopt a slightly compact structure. Also, there is a change in the hydrogen bonding network of the active site. Over a process of 2 stages, the phenylalanine molecule is converted to tyrosine, and since the active site of the enzyme has very low affinity for tyrosine, it is released from the enzyme. In the structural classification of proteins, or SCOP database, the phenylalanine hydroxylase enzyme has a fold number 2000896, a superfamily numbered 3001457, and a family numbered 4002591. The fold, superfamily, and the family have the same name, which is aromatic amino acid monooxygenases, catalytic, and oligomerization domains. This is a cartonic representation of the tetrameric phenylalanine hydroxylase enzyme, which I have created through PyMol. Here you can see the ferrous ions bound to the green subunits in blue and the ferrous ions bound to the red subunits in yellow. Here I have represented the mutation that I had talked about during the first slide. I have created this image through PyMol. Here we can see that the aspartic acid residue 415 has been changed to an asparagine. This is a part of the oligomerization domain of the protein, which extends from residue 411 to residue 452. Therefore, with this mutation, the protein cannot tetramerize, and therefore it cannot catalyze the conversion of phenylalanine to tyrosine. As a result, there is an increase in the concentration of phenylalanine in the blood, and this can lead to phenylketonuria. This is an electrostatic image of a dimeric phenylalanine hydroxylase. The red zones represent negative charges, the blue zones represent positive charges, and the white zone represents neutral areas. This image has also been created through PyMol. So that brings us to the end of our presentation. Thank you and have a good day.
Okay, thank you very much. That was really good also. A little bit long, but I think that you've got a lot of information covered in those slides. So very nice presentation, and hopefully we'll continue to talk about some of the topics that you mentioned there in today's session. Okay, so does anyone else have a presentation that they would like to share that is on here? Okay, so for those of you that joined later, we have reviewed a few presentations from over the weekend activity. The process is to continue working on your project, whether you stick to the same project or maybe you've discovered that you want to change direction, but we'll continue talking about this in our session today. Okay, so weekend activity. So again, last week, a lot of the focus was on the product of the gene, right? The proteins that actually perform certain functionality, and we've seen how a lot of the changes in the functionality are associated with disease. You've learned about the structure and the different physicochemical or biochemical properties of these proteins. And in today, or this whole week actually, we'll talk more about genomics and high throughput data, sequencing data that allows us to study how these mutations emerge and accumulate over time, including the human genes, as well as pathogens that affect what happens in the human host. Yeah, so for those of you that are still working on your presentation, you can definitely continue working on this presentation. Yeah, it's a draft, so we have shared the presentations that were recorded by video with Dr. Sona Vasudevan, and she will take a look at those and provide us with some feedback on those. For those of you that have not recorded the video, please record the video. If you can keep it fairly short, that would be great. And it doesn't have to be final. You can actually continue working on the presentation throughout this week. And hopefully by the end of the week, you will have a much more comprehensive project as a result of this whole program. Okay, so we'll talk today about how to do research literature review and how to design a research project. But we'll also talk this week about high throughput sequencing, next generation sequencing that allows us to detect mutations. And then once you already know where those mutations emerge and what genes and which proteins, you'll be able to associate how those mutations play a role in different types of conditions. So we'll talk about the process from the other side of the equation, right? You were kind of more focused in the first part of this program on the protein and the structure and the function, and we'll talk more about, okay, where does it actually start from and how do we get to the point that we know what are those mutations and what do they actually mean. So today we'll talk about a topic that will be kind of a transition, but in the meantime, we hope that you will continue looking through the online courses. I know that a lot of you have started. We have 2 courses that we hope you'll have time to complete this week at least, Introduction to Genomics and Genomics 1. Maybe some of you have already started. And there are additional courses like transcriptomics and metagenomics that you can, of course, explore to kind of refine your understanding about these different types of sequencing techniques. So in the context of understanding where the mutations occur, the different types of mutations, and understanding where what is the impact of those mutations in terms of expressed genes. The course access will not expire after the end of this week. You will have another week and a half or 2 access to the courses and the platform to finish up any pipelines. We do have a little glitch today that Joshua is actually working on to resolve. Some people have had issues with access. So if you're experiencing any kind of interruptions with access, we'll be fixing that very shortly and we'll let everybody know. But yeah, you will have some time to go through this. So the basis of the studies that we will continue to talk about is how to understand the impact of nucleotide mutations on the resulting amino acids, and how do we use the sequencing information that gives us a detailed review of nucleotide changes for different types of organisms, right, to understand how it applies to different conditions. So I understand that, you know, one of the examples that we talked about was sickle cell disease. Right, so I don't know if anybody actually chose sickle cell disease as your project, but as you probably heard, it relates to a specific mutation in the hemoglobin protein enzyme, and on PDB there's actually a molecule of the month. So they do these features about molecule of the month, and they choose different types of proteins or molecules, and they do a whole review. And I think it's a very fascinating topic, right? So for example, I don't know, maybe the majority of you already know this, but hemoglobin can change the way the blood appears, can appear red or purple or even blue. And the reason behind that is because oxygenated blood is bright red. So when you're cut, the blood that you see is very bright red, right? Because it's oxygenated blood. Deoxygenated blood is deep purple. So when you donate blood or give a blood sample at the doctor's office, right, when they look up in what came out of your you know, came out, it's not going to be bright red, right? So maybe you've wondered, you know, why is that? And the reason is because when you donate blood that is deoxygenated. So they specifically draw that kind of blood, and that's how it's stored. When you actually observe that in people, right, if it's not taken out, it actually looks blue. And the reason why it looks blue and not purple is because it is behind your skin. So that's just a different way that we can see that. And so some organisms like snails and crabs use copper to transport oxygen, and they really have blue blood. So some organisms actually have real blue blood. So hemoglobin is the protein that makes blood red. It is composed of 4 protein chains, 2 alpha chains and 2 beta chains, each with a ring-like heme group, or heme group, right? Hemoglobin, so heme, and that contains the iron atom. So right here, you can see a normal blood cell, and right here you can see a sickle cell, and this is the hemoglobin, and these red spots are those hemes. And here we even have a little animation that you can see with the iron atom. So when you see that oxygen binds reversibly to these iron atoms and is transported through blood. So in other words, you have ring-like heme group so this is the ring-like heme group and contains an iron atom inside. And oxygen binds reversibly to these iron atoms and is transported through blood. Each of the protein chains is similar in structure to myoglobin, the protein used to store oxygen in muscles and other tissues. However, the 4 chains of hemoglobin give it some extra advantage that allows it to pass this oxygen to other cells, right? So it's a fascinating machinery. The oxygen comes in through the lungs. It is passed on through the blood to all of the different organs in our body. And apart from oxygen transport, hemoglobin can bind and transport other molecules like nitric oxide and carbon monoxide. Nitric oxide affects the walls of blood vessels, causing them to relax. This reduces blood pressure. Recent studies have shown that the nitric oxide can bind to specific cysteine residues in hemoglobin and also to the irons in the heme group, as shown in this PDB entry. So the hemoglobin contributes to the regulation of blood pressure by distributing nitric oxide through blood. And the abuse of the heme group blocks normal oxygen binding and transport, suffocating the surrounding cells, right? So it's very critical. And sickle cell disease is caused by a mutation, and you can see that actually hemoglobin forms these fibers, right? And they could be terminated by this specific mutation. So as a result, the cells do not carry oxygen as well. But it is also, surprisingly, a genetic response to a pathogen. So malaria does not affect people with sickle cell disease as much as it affects people without sickle cell disease. So it's a balancing act, right? Evolutionary speaking, appearance of those mutations is a balancing act. In fact, the people that have this sickle cell gene that carry this mutation, they tend to live less, and they have trouble breathing in high altitudes, but they will almost always not be susceptible to malaria. And so when you think about where those people originate from, you can actually kind of make sense, right? So the logic is that wherever malaria was present, accumulation of sickle cell disease is prevalent, and wherever it was not prevalent, it did not occur. So as a result, we can see that there is a major interaction between the genome, or what we contain in the DNA, and the environment. The environment includes different environmental conditions like heat, etc., but also includes different pathogens that surround us. And to find the optimal balance between survival and being resistant to some of these conditions and diseases, we have an answer. And oftentimes that answer is to allow for some of those mutations that are beneficial to our survival to occur. So in this part of the program, in this second week of the program, we'll actually talk about how to study genomic mutations that lead to these kinds of conditions. And so we'll talk about how we can take next generation sequencing data that contains a lot of short reads, process that data, map it and align it to the reference genome, and identify genomic variants. So what the process that is called variant calling. And we'll discover how in different types of conditions, understanding what samples to use and how to compare them to each other or to what we have in the reference genome allows us to map those onto particular genes, like what you've done, but specifically looking at, for example, here is a gene called P53, and that gene has an accumulation of mutations that is typical in many different cancers. Around 40% of different cancers have a mutation in this P53 gene. And so we'll talk about how sequencing allows us to find and characterize those types of mutations. Now, interestingly, we will talk about both the challenges and the opportunities associated with these kinds of technologies. For example, here, you can see the alignment of short reads to the reference genome and specific positions that show a consistent change from what we find in the data based on our comparison with the genome where we believe it should be mapped to. These kinds of technologies have led to a major progress in the study of infectious diseases. Not only genetic disorders, like what some of the disorders that you have been looking at, but specifically in the understanding of how many different conditions, including infectious diseases like flu or coronavirus-caused COVID-19, or other typical infectious diseases, as well as many cancers. And so what a lot of research right now is being focused on the study of the interaction between different types of viruses that cause some of these cancers that are associated with different types of inflammation, really. And so we'll talk a little bit about these different conditions like Epstein-Barr virus that causes leukemia, human papilloma virus that causes HPV, or human papilloma virus that causes cervical cancer, and hepatitis B and C that causes liver cancer. So major pathogens that cause chronic condition first, and then they change and actually change the way that the human cells behave, turning them into tumor cells, as well as pathogens like the novel coronavirus SARS-CoV-2 that causes COVID-19. So we'll think about how do these different types of technologies allow us to study these conditions and understand the pathogens themselves, right? So what are these pathogens? Where did they originate from? How do they change over time? And then how can we find specific mutations and link them to the proteins that we understand their function, right? The challenge here is going to be that many of the proteins are not as characterized as well as some of the genes in the human body. But at the same time, we will talk about a number of different techniques to be able to characterize those genes and those proteins and look at their structures, which you already know how to do, to map the mutations from the genome onto the protein. So specifically, we'll talk about the spike glycoprotein. So who here knows which human protein does the novel coronavirus spike glycoprotein attach itself to? All right, excellent. Yes, ACE2 receptor. So we'll talk about, this is what you're seeing here. You are seeing here the spike glycoprotein from the virus attaching to a portion of the human ACE2 receptor. And the ACE2 receptor is also actually a complex. It's not just a single portion, and this is just one of its domains. So we'll also talk about the different functional domains and how do we characterize the different types of mutations as they start appearing in different domains. At the bottom level, kind of the foundation for these types of discoveries are not only our ability to know the structures, but many of them are actually linked to our ability to study sequences. And the study of sequences, here's an example of the original SARS-CoV-2 genome when it was published and assembled. You can see how it contains many short reads, and those short reads map onto a reference genome. And as a result, we see a consensus genome. And that consensus genome actually allows us to study in a very detailed way some aspects of more prevalent mutations. So here you can see actually the spike glycoprotein. Here you can see how those sequences end up in those FASTA files. And so we'll talk about how the difference between FASTA files, FASTQ files, and other types of file formats that are typical for the studies in infectious diseases as well as next generation sequencing in general can be used for different types of analyses. So by mapping those onto the reference genome, we can actually identify what function they play by looking at the product of that specific section, but it is not going to be as straightforward as it might sound as we start looking into this topic. Okay, so any questions about the assignment that you were given over the weekend? Maybe some of you had trouble finalizing your presentation. So if you have any questions, please let me know. Or if you have any questions about what we'll be covering in this week. Where is the assignment posted? I think the assignment was discussed in class for sure. Did we post it on the forum, Bibsha? I don't know, do you know?
The assignment, it is posted in the forum, plus I have also emailed everyone the assignment.
Oh, okay.
Okay.
Yeah, so the assignment was also emailed.
Yeah, and or I will email you once more the assignment. That would be fine.
Okay. Okay, so we will email you again about the assignment. And how did everyone record their presentation, Kylie is asking? Are you using Windows or a Mac, Kylie? A Mac. Well, on a Mac, I also use a Mac. So you can see in QuickTime, if it's a free thing that you should have. In QuickTime, what you can do is you can do File, New Screen Recording, and record that. So you just open your presentation, go through it. If you're using PowerPoint or Keynote, you can also record that. So actually, if you go into your Keynote, for example, and you want to record that, you can just go to play and then record slideshow. And that will allow you to go through all of your slides and actually record the sound and export it. So when you do File, Export to, here you can create a movie out of it. Okay, any other questions? And Megan is saying that she also used Zoom and made her own session recording. I'm trying to work on a gene of my interest, but I don't have any data related to OMIM or SCOP data coming in the Uniport ID, but there are a few papers I've read, and from them I figured this gene. Yeah, so we'll talk a little bit today about literature review and what you can extract, and also look at how different types of projects could be developed, right? So we'll talk a little bit about that Parimala. And if you have specific questions after that, that would be a good topic to discuss on the forum. Okay, yeah, so your question is how to study the activity if you don't have that information on a database? Yeah. Okay, we'll talk actually specifically about that topic on how to study protein function, and maybe that will give you some insights as to where to find that kind of data. You know, obviously I don't know what gene you're interested in, but we can, we'll definitely discuss that in the second week. Okay, so Muyua is saying that the videos disappeared. The videos are actually on YouTube. We cannot provide them in a downloadable format because the format, you know, these are not public videos, so we cannot allow you to download them. So please make sure that, you know, you're not sharing those necessarily. They are specifically for the participants of the program. And the reason is that because we have guest speakers, and those guest speakers are teaching at different universities, and of course universities actually pay them for that. So they are not supposed to be, you know, making all of their work public completely. Okay, so let's take a look at the forum here real quick. Okay, session recordings, let's see. So I think all of the recordings are here. Okay, so all of the recordings can be found right here. And unfortunately, like I said, we cannot make them downloadable. But you can definitely subscribe to our YouTube channel if you want to keep those links. Okay. Any other questions? All right. If there are no other questions, in today's session, I wanted to speak to you a little bit about literature review and project design. So the objective of this whole week is to take where you are right now with a specific topic of interest and develop it into a bioinformatic project. Now that bioinformatic project does not have to be about a single protein. It does not have to be about a specific element that you are studying. But it can be more general. And to give you a little more understanding of what I mean, I would love to give you a few examples in this session. So we hope that the outcome of this whole week, or in today's session, hopefully you will learn about different types of data sets and different types of repositories where you can search for that kind of data. But overall, hopefully in the next few sessions, you'll be able to develop the research question that you have, and then find the right data to be able to answer that question. So that is really the objective of this whole week. And then at the end of this week, you'll be able to submit your final project. Let's say some of you have these great slides as an introduction, and those slides are really just literature review at this moment, right? So we'll talk today about how to take those further and how to actually incorporate some data analysis into your project submission. So the different types of data that you might have looked at so far, you probably see that different topics have different number of publications and data sets. So here, for example, if you look for sickle cell on PubMed, which is the repository for all of the publications on NIH, on NCBI, National Center for Biotechnology Information, you will see that by looking at sickle cell, you will find 1,000 or almost 2,000 publications. Or sorry, in 1939, there was one publication, right? So you can see that those publications started a long time ago, starting from 1923, and have increased over time, including a number of publications in 2020 that is actually less than in 2019, right? But that is because we're just in the middle of the year. So as you can see that overall, in the research project is just a continuation of our weekend activity where we build on what we've already searched. So you're not limited to just that one topic. That was an example to get you started. So you now know how to research proteins, you know how to explain what's going on, kind of provide a background for what you've researched. you're still able to change that if you want to focus on something else. And hopefully you can start working on that today. But I'll talk about how a research project is developed. I think that given the amount of time that we have, if you're able to use where you started, that's probably going to provide you with more time to study different other elements, because we'll talk about what are the unanswered questions and how can you contribute to some of those unanswered questions. Okay, does that answer your, okay, great. So for example, here, right, what I was trying to say is that sickle cell has been studied since the 1920s. However, even today in 2020, we probably see that there will be more publications than in the previous year. And that means, right, so here you can see 30,000 publications on sickle cell, and seemingly we know everything there is to know about sickle cell, right? You already did a study, you see the protein, and we know what its function, and we know what mutation is causing the problem, but nevertheless, research continues. So just look at some of these top recent publications. CRISPR-Cas9 for sickle cell disease, applications, future possibilities and challenges, microparticles in sickle cell disease. So the diseases that are described and taught in a textbook are typically an oversimplification of the real condition that is observed in real life. And so the research continues. Here you can see for coronavirus, again, the first publication was back in the 1940s, but you can see in 2020 a huge spike in how many are actually there. You can see that there's a total of about 37,391 results in total. So how many were in 2020? 22,131. So practically the, what would it be? 2/3 of all of the publications about coronaviruses were in 2020. Okay, look over here, some of these top recent publications: host factors in coronavirus replication, broad spectrum coronavirus antiviral drug discovery, coronavirus envelope protein current knowledge, right? So every single topic, what would this be? This would be a basic research in microbiology. Here you can see a drug discovery. Here you can see a specific protein that is still being researched, right? So the term coronavirus, which yields closer to 40,000 results, and the majority of those in 2020. How about diabetes? This is a topic that yields 51,789 in the year 2019 alone, which means that it is more than both coronavirus and more than sickle cell disease. And all of that was just in 1 year, right? So you can see how much it has been studied. The first studies appear back in 1788, and a total you have of over 766,000 results. So here you can see a number of different types of publications and a number of different types of results that they yield, right? So when we think about some of these topics, no matter what topic it is that you think has been studied extensively or is just occurring today, like this novel coronavirus, the amount of publications is continuing to grow very steadily. Here you can see if you actually select publications with associated data, the number is decreased dramatically, right? So when we talk about publications, they are not necessarily publications that have produced some new data. And that's an important part of it, right? So if you limit this to only publications with associated data, look at how much less we have. In 2019, there were only 38 publications with new data about sickle cell disease. For coronavirus, in 2020, there were only 158 publications with new data. In diabetes, in 2019, there were only about 2,000 publications with new data. Still dramatically more than these other two, but you can see how the numbers are going down a lot more. Okay, so let me ask you a few questions. We'll look at a number of publications, and I will ask you what type of a publication this is, because there are a number of different publications. There are review publications, there are different types of opinion publications, and there are primary research publications that actually produce new data and provide some new insight into what their topic is. So let's take a look at some of these examples, and I will ask you, tell me whether this is a research publication that yielded new data, or is this a review or an opinion publication that did not produce any new data, okay? So one or two. The one produced new data, gave some new experimental results, or two is did not produce any new data, but it did either new analysis or new opinion. So here's the first article. It says SARS-CoV-2 specific T-cell immunity in cases of COVID-19 and SARS and uninfected controls. What do you think? Look at the this abstract right here. This is the abstract, and look at one of these figures from the publication. So tell me, did this publication produce any new data? Okay, yes. Okay, so one, it did produce new data. Two, it is an opinion. It's a review. It's a meta-analysis. Okay, great. So Parimala, you're saying two, and Karina is saying two. So you're thinking it did not produce any new data, right? So let's read this a little bit more and understand what is it that they actually produce. So memory T cells induced by previous pathogens can shape the susceptibility to and clinical severity of subsequent infections. Little is known about the presence of pre-existing memory T cells in humans with the potential to recognize SARS-CoV-2. Here we first studied T cell responses to structural nucleocapsid protein NP and non-structural NSP7 and NSP13 of ORF1 regions of SARS-CoV-2 in COVID-19 convalescence n equals 36. In all of them, we demonstrated the presence of CD4 and CD8 T cells recognizing multiple regions of the NP protein. We then showed that SARS recovered patients, n equals 23, still possess long-lasting memory T cells reactive to SARS NP17 years after the 2003 outbreak, which displayed robust cross-reactivity to SARS-CoV-2 NP. Surprisingly, we also frequently detected SARS-CoV-2 specific T cells in individuals with no history of SARS, COVID-19, or contact with SARS-CoV-19 patients. Okay, so Parimala, do you think that they did produce new data or they did not produce new data? Okay, so actually, yeah, they did produce new data, right? And here they actually describe, so whenever they describe, they took specific patients, right? So they did produce new data because they describe here how they took these patients, right? And they actually studied them for T cell memory, right? So this is an article that did produce new data, and they actually describe this new data right here, right? So this is what's called raw data. It has original data that they describe, how it was produced, which patients it was taken from, and what was their analysis, including what their conclusions or what their findings were. Okay, so let's take a look at this, another example. Emerging SARS-CoV-2 mutation hotspots include a novel RNA-dependent RNA polymerase variant. Okay, so here you see SARS-CoV-2 is RNA coronavirus responsible for the pandemic of the severe acute respiratory syndrome, COVID-19, RNA viruses, etc., right? So here, take a look at some of this description. Tell me, did they produce new data or did they not produce new data? So 1, they did produce new data. 2, they did not produce new data. They just analyzed somebody else's data, or they provide an opinion on somebody else's data. Okay. Okay. So no new data, or 2 so far. Excellent, right? How do we know? So they didn't because they didn't conduct an experiment of their own. Right. So let's take a look. Right here, you can see data availability statement. The data sets analyzed during the current study are available in GISA8 and GenBank repositories. Right. So here you can see. Right. So here you can see that they analyzed 220 genomic sequences from GISA database derived from patients infected by SARS-CoV-2 worldwide from December 2018 to mid-March 2020. And they used the SARS-CoV-2 reference genome that they obtained from the GenBank database, right? So as you can see here, as opposed to the previous, when they dealt with the patients and they collected samples from the patients, here they took data that was already deposited on GISA, which is a database, and they used a reference genome, again, from a different database. So here you can see their conclusions. Article is something they made from what was published or experimented separately from what they might have written an article. So Parimala, if you could please change the chat from private to public, we can all see. So yeah, the difference I'm trying to point out is that the people in the previous article, right? So in this article, they actually took samples from patients, okay? So they dealt directly with patients, produced new data, and here you can see what they did. They were, so look, 9 COVID-19 recovered individuals were stimulated with 12 different pools of 7-8 NP peptides. The table shows interferon gamma, ELISA spot response against the individual NP peptide pools. So they described the experiment, right? So they described the experiment, they describe how they collected data, and they provide some findings, right? A lot of times, the difference is that in these experimental papers, you are producing novel data and maybe not conclusive insights, right? So even though this gives us a hint of what might happen with these T cells, it is not a study of thousands of people to really understand what's going on, right? So they only had 36 plus 23. So it looks like, and maybe 37 took 220 genomic sequences, so the number might be greater, but they did not collect that data themselves. They actually took data from all over the world and they analyzed that data, right? So this is an analysis paper. It's not, does not provide us with any new data, okay? So it's new analysis for existing data. Let's take a look at this other one. So here, genomic variants of the 2019 novel coronavirus or nCoV coronavirus. What do you think this is? Is this new data or is this analysis of existing data? Okay. All right, excellent. Yeah, so this is an analysis of existing data. How do we know? Right here, they are saying, taking advantage of all the genomic information currently available, we constructed a phylogenetic tree, right? So when they describe the paper, when they describe what they did, they are describing the analysis. Okay, so Karina is asking, so are you just speaking of data if the origin of the data is a new experiment? An analysis then doesn't provide new data because it is based on existing data. Exactly, new data. That doesn't mean that it doesn't provide you new information, okay? New information is the result of analysis, but it does not produce any new data. The data was already there, collected by somebody else. Okay, does that answer your question? Okay, and that's important. Why? Because, I mean, let's take a look again at some of these numbers. Okay, so here you have a total of 30,000 results about sickle cell. You have almost 40,000 for coronavirus. Here you have 766 papers, right? These are publications about diabetes. But if you reduce the number for only associated data, you can see that the numbers are much less, right? The total number for papers with data, with associated data, for sickle cell, 298, coronavirus, 949, diabetes, 13,947. So a small proportion actually provides you with access to new data. If a paper does not provide you new data, it's not going to show up. Okay, so here are some examples, right? You correctly identified what types of publications these are. And the reason why this is important is because when you are asking a question on one side, right, if you are not able to generate your own data, you can still do a lot. You can still do analysis. You can do comparison. You can come to your own conclusions. There's a lot more that you can do than just generate a new data that will provide new insight, right? The new insight, the new information that your analysis shows could be based on somebody else's data. Okay, so this is new analysis. Let's take a look at this, okay, and try to understand what kind of a publication this is. What do you think? Are they providing us with new data? Are they describing new data that they generated in this publication? So one, they generated new data. Two, they did not generate new data. All right, excellent. So why do we know? How do we know? Let's take a look. So this is actually correspondence, right? It says right here that this is correspondence. And two, they are saying here that they are providing a new perspective, right? So they are saying here, we offer a perspective on the notable features of the SARS-CoV-2 genome and discuss scenarios by which they could have arisen. Our analyses clearly show that SARS-CoV-2 is not a laboratory construct or a purposefully manipulated virus. So again, here they are doing some novel analysis that provides an important perspective. Okay, and the reason I am highlighting these is because they provide very different types of insights. In all of them, they did some analysis. In all of them, they highlight a specific topic, but which one it is and how they arrived at that conclusion is very different. So when we are thinking about choosing a topic for your project, okay, there are a number of considerations for you to think about. The first one is you should select a topic that you know and care about. So when you select a topic for a research project, you should ask yourself, can you prepare a few slides explaining the topic background, right? So as you found, some of them are more difficult than others. Some of them you care less than others. So for example, can you see yourself spending a few months researching this topic? If you don't see yourself continuing and being motivated by researching this topic, even when issues arise, like there's not a lot of data available, or maybe you cannot find exactly all the information in one place, you have to collect it from multiple different places, that should be an answer for you whether that topic is the topic for you. So find a topic that you know and you care about. The other side of it is what will be interesting to your audience. So sometimes when we think of a topic that is very niche, it is very specific to a small group of people, and that group of people is your audience, that is fine. Sometimes you have a group of people that you are trying to show this to that has no interest in what you're trying to say, right? So in your case, it's a little bit less defined because your audience is very general right now. But because your audience is general, you should probably take a project that has some relevance to the general audience that you're going to be presenting to. The other, so yeah, how do you know? You can ask yourself, are there enough people who care about the topic? Or do you hear questions or discussions about this topic, right? Because the general audience is going to be probably most interested in the topics that are widely discussed and are on everybody's mind, right? So when you think about those topics, right now, a good example is COVID-19. That doesn't mean that I'm telling you to do your topic on COVID-19, but I'm just saying that, you know, there are only so many topics that have a wide audience appeal. And another one is that sufficiently studied and data available. So you can look at the number of publications, you can look at the number of publications with data. So you know that the topic that you've picked actually has been studied sufficiently and has enough available data. For example, are there publications and reference studies on this topic? And are there any data sets to study this topic in detail? And we'll talk about how to find some of those and answer some of those questions. So the intersection between these three is where you should focus on a topic for your research project. Okay, so the other thing that you should think about, because you're probably not going to be generating any new data in the context of this research project, you should think about both how you will conduct your research and how the papers that you are going to review have been published. Okay, so there are three types of research: primary, secondary, or tertiary. So primary papers are the ones that produce raw original data, and they describe firsthand observations. The secondary is the analysis and interpretation. It does not provide new evidence, but it's more of a review of what has already been generated. And then tertiary might be compilation, databases, data tools, and collections. All three of these are very valuable. It does not speak about the value. It just talks about how we view these different publications in terms of, let's talk first about how they are published. So how they are published, somebody, sorry, wrong direction. So somebody identified a problem or a question, and that problem had to have data about it, right? That question had to be answered with research data. So they had to first produce that data, then they had to analyze that data, and then they had to make it available, describing the methods and the tools that they used for it. And at the end, they arrive at a solution or an answer. When we do literature review or researching a topic, we're actually going the other way around, right? We most likely will first start from some kind of a database or a collection. We will then review analysis and interpretation that other people have done. We will think about what was interesting, what kinds of questions were being asked, what kinds of interesting topics were selected in the context of this particular main kind of a general topic. And then eventually what you want to do is you want to review some of the available data about that specific topic and arrive at a problem or a question. So you actually want to generate a question by going the other direction. So when we think about preparing that project, you typically start with some kind of a general background overview, kind of similar to what you guys have already done, right? You've researched some databases, you've found some interesting insights, you've been able to narrow it down, and now maybe you're starting to think of a more specific question or a hypothesis that you would like to propose. So typically that starts from first reviewing available sources and categorizing them, okay? So for example, many people say this and many people say that, and there's a group of people that say this, and so you're really organizing and grouping the different publications or the different data sets into groups and to define distinct groups of knowledge. Then you typically want to identify gaps and unanswered questions. Maybe pick a controversy or a debate. You might say, this group has proposed this, another group has hypothesized something else. You see that maybe they do not agree 100% and there are many different views. as a result of spotting that as an opportunity, you will want to identify your own question or hypothesis. Okay, so for example, in terms of COVID-19, where we have rapid acceleration of publications and data sets being available, you might want to say, some have proposed that this specific mutation that accumulates in different populations has a significance for pathogenicity. On the other side, there is this group of reviewers or scientists that believe that this is a random mutation that has occurred and has no specific pathogenicity as a result. How might you answer that question? Now you have to start looking for a specific data set that might provide you with evidence, and that evidence will be the answer, right? If you provide another opinion that might be valuable, if you provide another method to study this question, that might be valuable as well. But ultimately, if you are going to answer that research question, you need to answer that with data, with evidence that shows what the answer is. Okay, let's take a look at another example. We'll put aside COVID, and let's take a look at the specific key terms. And this is a way for you to think about a data set and a publication that you want to read to know that you have understood it, right? So here's a publication called Modeling Precision Treatment of Breast Cancer. And you can see highlighted are some general terms. As you read through this publication, it has to be clear to you what each one of these terms mean, okay? So let me ask you, what do you think are molecular profiles? Okay, Paramala is saying structure. Anyone else? Molecular basis of a disease, sequences, or sequence data, gene expression data, and more. Okay, so I would say the closest is probably what Suparno is saying, right? Molecular profile is the molecular basis of a disease that is evident in different types of data. So for example, as you see this visual abstract, you can see that they actually used a variety of data sets, okay? And Emeka is saying molecular signature. A signature is a recognizable pattern, right? So that pattern is the basis at the molecular level of some phenotype that we call a disease. Okay, let's look over here in the second highlight, identification of features. So what are the specific features that we would like to identify? What features are there from these different molecular profiles? Maybe give me an example. Okay, mutations, that's a good example. What other types of features could be molecular features? Genes, okay, what else? Epitopes, ligands, okay. Epitopes are essentially kind of a pattern, so yes, gene expression. Okay, so levels of gene expression. Modifications of proteins or promoter regions mentioned. So modifications, right, that's the key word, is that the features are the specific modifications. So let's take a look, what are those specific modifications? You can see here, levels of expression, right? So different levels of genes that are being expressed. Individual mutations, right here you can see a mutation. So those mutations are changes, right? They're changes in a sequence. And what sequence? That is kind of the next question. They could be copy number variation. So a gene that occurs more times or less times. They could be specific patterns of chromatin immunoprecipitation, where we see specific changes associated with chromatin changes, right? So those are called molecular changes as a whole. So we are discussing here how molecular profiles, right, let's call them sentences, that we use to describe these specific patients are specifically defined by the order of letters, by the length of words, by the order of the different punctuation symbols, etc. So we are actually looking at sequence data, and we can identify specific patterns, and those patterns can be composed of specific features. And here we see the objective, right? Can predict therapeutic response. So in the context of this paper, what they are proposing is a method of analysis, right? So here you can see that they are proposing a very specific method or an algorithm that can help enable to identify molecular features. Again, we talked about what those features are, associated with responses for different types of breast cancer cell lines. Okay, so again, it's important when you read, when you do your literature review, you need to understand what kind of literature am I reviewing? Am I reviewing an opinion? Am I reviewing an analysis? Am I reviewing some primary raw data that somebody generated? And you have to make sure that you understand each one of the terms, that as you're reading through the paper, it makes sense, so that your review yields a specific conclusion. So what might be a conclusion from literature review? So again, the conclusion from literature review is that you understand the broad context of whatever topic you are interested in. If you're interested in cancer, maybe it's a specific type of cancer, maybe it's a specific type of patients, maybe it's a specific type of an intervention strategy. And as you start narrowing that down to a very well-defined question, you actually want to start thinking, how do I then identify an area in this question that I can address? Okay, and then your address of that question, your answer to that question becomes your solution, becomes your project. Are there any questions about this? I am still struggling to see how we could contribute. Okay, well, let's see. So the first project step is to frame the problem, okay? Oftentimes, that is the most difficult part of your project, is to really frame the problem and identify a very specific focus for your project. Because when we just say sickle cell disease, I mean, there are 40,000 papers on sickle cell disease, so you might be just doing literature review. If you want to do a research project, you have to be able to collect the raw data needed to answer a very particular focused question about that general topic. And then you have to communicate the results of your analysis, showing what conclusions have you arrived at. How can you contribute? That depends on the topic that you've selected. Some topics have a lot of available data. Other topics don't have that much data. You want to make sure that you select a topic that has sufficient data for you to do some kind of analysis. Take a topic that is motivating to you, because whatever these steps are, they're going to include struggles. They're not going to be easy to address. So you want to select something that you are motivated enough to address to actually come to some specific conclusions. Okay, so Karina, does that answer your question, or is there a specific topic that you're interested in? Okay, so I'll give a few examples, right? I'll give you a few examples of how we could actually apply some of the things that we are learning about in this program to develop a problem. So let's talk about that first stage, framing the problem. How do I search specifically papers on PubMed? Okay, I will show that in a second. So framing the problem. There are many different types of problems. They could start from a clinical problem, they could be an industry-specific biomedical problem, or they could be a research problem. So these problems could be mechanistic studies. Okay, who knows what is a mechanistic study? Does anyone know what is a mechanistic study? Exactly. So Parno, studying the mechanism, right? So if we are focused on a specific protein, let's say, and it has a function, we can study that mechanism. If we are looking at a specific, exactly, trying to detect mechanism of action, for example, we want to study how the virus enters the human cell and replicates, right? That's a mechanistic study. We want to understand basically the mechanism at which something happens. Okay, principles, they could be principles, not the mechanism, but principles, more general. For example, you see here principles of evolution, adaptation, and replication, right? So we don't want to focus on a specific mechanism, we want to understand the principles. For example, evolution in general is higher, or the rate of mutation, right, that evolutionary mutations are actually more prevalent or occur at a faster rate for this particular group of viruses as opposed to those groups of viruses. Okay, comparative analysis. Comparative analysis means that we have one type of a virus and another type of virus. We want to compare them. How are they different? Or we have this type of a cancer and that type of a cancer. How are they different, right? How are they causing different interaction between different elements in whatever topic we are selecting? Transmission. So how does the disease spread? And so we can really look at the basic research questions that are answered for research purposes, right? Just to generate knowledge. We want to understand and really be able to study this process. Sometimes that leads us to an application, right? So clinical or a biotech application would include, how do we use this data? What do we use this data for? How do we find the correct types of patients? How do we diagnose disease accurately? How do we identify people that will respond to therapy? How do we actually take this use of the mechanistic understanding of disease progression or the disease occurrence to be able to do something about that, okay? And in terms of creating those interventions, especially if they are vaccines or drugs, how do we actually apply this knowledge to be able to make new drugs, make those drugs available to patients, and make those drugs given in a more efficient way. Okay, so a lot of times, how does this work? A lot of times, and I will actually show you, we have some project examples. Once you read a paper, most of the time, that paper will refer to some kind of data and some kinds of methods of analysis. So this is published and digested. And these are excellent examples that we can start with, and we can start looking at the data, and we can understand how the data will actually be analyzed by somebody who is already an expert in the field. And then once you look at this example, right, and maybe you've chosen several of these examples, several different papers, several different approaches that have been applied to some kind of type of data, you take that data and the methods, and you can actually add on to those from the data that you've discovered elsewhere or from additional methods that you have found elsewhere, and design your novel and independent project, right? So you're not replicating exactly what somebody else did. You are basing it off of these examples, but then you are actually looking for a new type of a project that has some variation to what has already been done. So the first step is to collect this raw data needed for your problem. Okay, so let's take a look at some of the places where you can actually do that. And I'll start from NCBI, where you can start doing your literature review. Okay, so in NCBI, let's make this a little bit bigger. You have here all your databases, but you can look through specific databases, and this is a very comprehensive repository, right? There are some alternatives, but you can actually look here as a great start. And the first one is PubMed, right? So right here, if you select on PubMed, you can search for, let's say, sickle cell. Okay, and this is what you see. Let me make this a little bit more. This. Okay, so you can see here the specific papers, and you can also organize those into different types of articles or specific publication dates, or look at additional filters that they have. Okay, so for example, you can actually look at clinical studies or clinical trials if you're interested in clinical applications. You can look at observational study. You can look at, let's see, comparative study. You can look at editorial, right? etc. So you can really look at a number of different types of literature as you search through. And the important part is to start from some general topic that you're interested in. Are you interested in cancer? Are you interested in infectious diseases? Are you interested in some specific type of an organism that maybe you've been fascinated by? That's where you should start with, right? So start with here, and then start going through the data, going through the publications, looking through the different methods of filtering through, right? And coming up with a broad description that you can start narrowing down. If you want to find data sets that have, or yes, data sets or publications that will have data sets, right? Because you want to find ultimately some data to work with, as this is a bioinformatics project, you might want to find some associated data. And then this data could be of very many different types. And in fact, if you don't go for PubMed, but you go back to NCBI, there are specific databases on NCBI that you can start looking through for a very specific type of data. For example, you have bioprojects, which are collections of data sets. You have, for example, genomes. You have gene expression omnibus data sets. These are data sets that speak about expression of genes. Okay, you have nucleotide sequences. You have protein sequences, SNPs, raw FASTQ files that have individual reads and could be processed to analyze in different ways. So for example, let's take a look at SRA. SRA will provide us with a variety of different types of sequencing data. So SRA stands for Sequence Read Archive. And this is high throughput sequencing. You can see that all of them are Illumina platform. So if we look here, we have a number of different types of data. Data that is about DNA variation or RNA. Data that is controlled means that you have to request access, or public. You don't have to request access. You have here exome or genome sequencing, paired or single end, etc. Right? So you can start looking at all of these, and of course you can clear all of these. I believe I put in here Illumina. No, maybe not. So, and also you can look through here how many of the different data sets are available. So if I look here for sickle cell and I find this first result, I can study what it will include. And a lot of times it will include an abstract that will tell me what exactly they were studying, right? So this is Staphylococcus aureus, sequence type 72 is prevalent human pathogen in South Korea, which lacks Panton Valentine leukocidin. It has been shown to possess resistance against oxacillin, erythromycin, clindamycin, tetracycline, etc. So a lot of different drugs. And so this is a drug-resistant pathogen. And if you want sequencing about that pathogen, maybe to study the accumulation of mutations that cause it to be resistant to drugs, you can find it right here. If you want this individual sequence, just one sequence, you can click here. If you want to see all of the sequences, you will go to the project. So the bio project. This is the bio project. The whole bio project actually only has one biosample, but they have other types of sequences that probably have more, right? Here's an example of RNA-seq, okay? And it's probably because this is ex vivo anti-sickling activity in human erythroid cells, right? So this gene therapy reduces sickle hemoglobin production, allowing for ex vivo anti-sickling activity in human and erythroid cells. And if you go from here to the bio project, you will find probably more data sets. Okay, so here we have 6 SRA experiments, and they are probably some kind of a comparison. Okay, so let me ask you, what might be a topic of interest, right? Does anyone have a topic of interest, just so that we can take a look and see whether we can find some data? Okay, great. So gut dysbiosis. So on all of gut dysbiosis, you can look through different types of data sets that are available. Gene expression data sets. You can look at bioprojects, right? You can look at nucleotide sequences, sequence read archives. So you have a lot of different places you might want to go. You can also look on PubMed. So let's just look on PubMed and find associated data sets. So here you can see stress ulcer prophylaxis versus placebo, a blinded randomized control trial to evaluate the safety of 2 strategies in critically ill infants with congenital heart disease. Impact of dietary propionate on fructose-induced changes in lipid metabolism, gut microbiota, and short-chain fatty acids in mice. Comparison of the gut microbiome composition among individuals with acute or long-standing spinal cord injury versus able-bodied control, right? So for example, spinal cord injury and gut microbiome, right? That seems to me like an interesting study. So here you can see what they were trying to do. And they were looking at alpha diversity or composition of the microbiome. So they were really looking at generating preliminary data and a scientific foundation for future studies investigating the impact of the gut microbiome composition on long-term health in individuals with SCI, which is acute spinal cord injury or spinal cord injury in general, right? So now if you look here, you will have a link out more resources. Let's see if they have the data. Well, let's go look over here. Okay, so this is behind a paywall. But if you look for this specific name and look for it on NCBI, you see they do have information about it, but it is protected. So dbGaP, you need to ask for access because it's probably generated from specific patients. But if you're interested in this, you could probably look for gut microbiome and acute or just spinal cord injury. Okay, and here you have, for example, gene expression omnibus. You have also SRA studies. So let's take a look at SRA studies. So you can see these are microbiome samples. Illumina MySeq is typically microbiome. We'll talk about the different types of data that we can find here and how to analyze them in some of our next lectures. But an example that Chandrani here asked, just a topic for gut microbiome spinal cord injury or gut dysbiosis. Let's say her general interest was gut dysbiosis. We then looked through publications, and let's say I'm just imagining I found it interesting that we have this kind of injury in the spinal cord related to gut microbiome. I start getting interested. It looks here like this is a fairly new publication, right? So it was published in June 2020, so this month. And they're saying that this is going to provide some preliminary data for people to continue studying this topic. So it seems like an interesting topic, and people are studying it now, currently. So, and here we have some data, right? We have some available data. What kind of data do we have, and what kinds of comparisons maybe we can make? So let's take a look. This is maybe only 3 different data sets. Okay, so there you can see transcriptional changes, activity-induced changes in the liver transcriptome, spinal cord injury, and gut microbiota. So this looks like the data set we're interested in. We have 23 different samples, right? So this might be an interesting study that I can propose, right? So I can propose some kind of an analysis here to see what I can find. Okay, so typically, like I mentioned, we have a publication. The publication will reference some type of data, some number of data sets that we can analyze. They will describe some methods, and we of course have to, and we will throughout this week, understand how some of these analyses are performed. Then we will apply this to potentially new type of data, maybe modify the methods that we have, maybe do something differently in terms of methodology, or doing something different in terms of the data, maybe apply the same methodology to a different data set. And then we can actually develop our own novel and independent project. Okay, so Mecca is asking, is it okay to combine projects, say gene expression in sickle cell patients, with a particular phenotype, say hemolysis, and those in thalassemia patients with same hemolysis to understand mechanism of action? Yeah, so I think that, you know, again, I'm not an expert on sickle cell disease, but, you know, if we have a particular subgroup of patients with sickle cell disease, right? So that would be that particular phenotype. And then you want to understand how different is the mechanism of action, you know, for a particular development of that disease in different patient groups. You know, that's an excellent type of a project that relies on data that has been already generated. And that might be a hypothesis generating, right? You're doing your analysis to generate a clear understanding of what might be the mechanism of action. And then you are also developing it to be able to understand how to actually analyze this if you had additional data that you potentially don't have. Okay, any suggestions if the disease has multiple factors or protein or pathways that cause it, and not just one mutation like sickle cell anemia? How to approach or go with such disease in making a project? Well, in fact, I think the majority of the diseases have multiple factors. And more we are realizing that, as there's not many examples like sickle cell where it's one gene or one mutation related to just one phenotype. There's a lot of variation to the phenotype, and there are a lot of associated features with that one particular mutation that you might be most aware of. So in many of these that we will talk throughout the next few days, we will be talking about kind of hypothesis-free and understanding at the level of data-driven. So we'll look at, for example, gene expression data. Gene expression data will tell you a lot about many genes, and in all of that data, you will have to find specific patterns that actually address the question that you have. We will talk about viral mutations in viral genomes. And typically there are many mutations, not just one. In fact, a lot of them, thousands. And we'll talk about, okay, well, how do we select the ones that are associated with a phenotype? Or how do we select the ones that might contribute to a certain mechanism? We'll talk about also finding different types of microbiome data and metagenomic data. So we'll look at how does composition, and composition means not just one type of bacteria, but many types of bacteria organized into a group, maybe by their taxonomy, that cause some kind of a result, a phenotype. Okay? So I think that's a very typical situation, how to do it. We, of course, will have to discuss those methods of analysis. So how do we go about it after we identified that specific topic? We have to then go and collect the data, and we already looked at that. So let's look, there are not only specific data types, like we saw now, gene expression, metagenomic sequencing, genomic sequencing, etc. There are also specific types of dedicated repositories. For example, for virology, there's a new database called NCBI Virus that allows us to study genomes of many different viruses and even explore them visually so that we can understand where to focus our questions, easily select the types of comparisons, the types of conditions. And there are data sets like GIS-AID that we also kind of briefly talked about, where a lot of these sequences are already analyzed and annotated, and we can leverage that annotation and analysis to generate a hypothesis of our own. Once we've identified that question, and we have done our literature review, and we have started collecting that data, it's important for us to understand the process of analysis. Typically, analysis is organized into at least 4 different steps. You first process your data. That might include finding and annotating the different sources of data, making sure that the data is cleaned up, mapped, or somehow annotated with a reference, and maybe quantified. So you have done your data processing at that stage. Then you explore your data. You explore it for statistical properties, maybe visually summarizing that data, and potentially normalizing, making sure that it is in the same scale. You're comparing apples to apples and not apples to oranges. You're not dealing with different types of measurements. Once you have done and prepared your data, you then analyze that data, testing your hypothesis, maybe mining the data for different patterns, or even applying machine learning to understand what this data can train in terms of the model that you want to prepare. And then at the end, you want to annotate and make sure that the results of your analysis correspond with some biology that could be interpreted. You can actually take a look at the patterns or the statistical characteristic of the data and provide with a biological or a clinical interpretation. So to help you with this process, which I understand is a lot, and it requires some kind of a effort to start thinking about the specifics of your project, we actually provide you with several examples. And here are some examples in cancer. We also have some other examples that are not cancer related. And these provide you with a good starting point. If you want to, for example, look into one of these projects to understand what type of data they have and what types of insights this data can provide with the right kind of analysis, then you might prepare a presentation, and that presentation being able to speak about your project, being able to answer questions is a critical part of that process. So for example, over the weekend, you've done some literature review and you've identified some specific examples that you were able to present in a few slides. What we would like you to do next is start thinking about how do you actually present a result, not just of a process that you did, but of a research study that you have conducted. And that research study requires you to be able to answer questions, to be able to rightly reference the sources of the data, and to be able to provide a focus for your viewers. So many times, these are going to be, let's say, posters in the end, or the project presentation could be virtual poster presentation. In that poster presentation, you have to identify specific portions of your project. For example, an introduction, kind of similar to what you have done over the weekend. Methods, and I have here an example. Yeah. So introduction, methods, the results of your study, and the conclusions that you have drawn from this analysis. Okay, so as we go through the next several steps, through the next several days of these meetings, think about how they relate to the project that you would like to work on. So these are examples from some previous participants of our programs, and you can see that you can work on your own or potentially work together with others in a group. It is always beneficial to work together in a group because you have more time to work on your project. Your project can be more comprehensive because you can try more things, and you can probably divide up the tasks like data collection, data analysis, biological interpretation, and even the process of presentation that each one in your group might be more familiar or more comfortable with, so that overall your project is graded, or the result is overall between all of you will have a bigger impact. Okay, so in our next session, so we will continue to work on our projects after this genomics course has ended. At the end of, so let me walk you through some of the coming up sessions so that you can kind of think, how can they help you develop this project proposal? In our next session, we will talk about infectious diseases and genomics in virology. So we'll talk about how we can identify viral species and strains, do comparative analysis of viral genomes and proteins, study viral diversity and specific mutations associated with disease pathology. So you will learn about next generation sequencing and how this type of data from next generation sequencing could be used to study viruses. In the session after that, we will talk about next generation sequencing in microbial communities or in the microbiome. You will see how we can use specific genes like the 16S ribosomal RNA or specific hypervariable regions in those genes to understand microbial diversity and study its composition. And we will learn and understand how metagenomics can be studied directly from samples without culturing the bacteria in the lab. So you will now get some experience with analyzing viral genes or viral genomes, metagenomic data, bacterial data, and study its composition and associate those with human disease. Then we will talk about how vaccines, antibiotics, and antiviral drugs could be studied. For example, specific differences between bacterial and human metabolic pathways, the process of viral cell entry and replication, and how the host immune system and the evasion of the immune response could be studied using these types of data sets. So that will give you some insight into intervention, how the drugs or the vaccines could be used to treat infectious diseases or diseases caused by viruses and bacteria. So this will probably provide you with a specific focus already, right? So up until now, we've kind of covered more of the human genetic diseases and disorders. Now we're going to talk about non-human pathogens, which are non-human. They're pathogens that affect the humans and cause human disease. but we can look at specific types of data that can characterize the basis of that disease. And we can understand the treatment part of it as well. So during that time, we will actually look at specific data sets, run some analyses, and see the outputs. In the context of that process, you will primarily work with NCBI virus because those sequences are much shorter and much faster to analyze. That doesn't mean that the analysis is simple, but they are faster to analyze. So we are hoping that some of your analysis ideas are going to be centered around viral or bacterial caused disease. We hope that you will be able to find a data set that is not too big so that you can do your analysis even at a preliminary rate for the next weekend, right? So the goal is to have a project that could be presented just after this week to a panel of judges. Your total project presentation will have no more than 10 slides, and you will either take one of the conditions discussed so far or any project of your own. It's not limited to the projects that we have already discussed, using some of the methods and the data that you have learned about, and then you will upload your project just like you did with the video or with the slides, and we will have judges that will provide you with personal feedback to the project, and obviously there will be a score for each project, and the top project will actually be highlighted. So we will highlight your project, we will include your personal journey, how you have learned all of these different things and what you plan to do with it in the future. We will include your project presentation, any kind of interaction that we've had, and then the judges will look at all that together as well. And after that, right, can you continue to work on your project? So for some of you, that might be the end, right? That's all you want to do is work for the 2 weeks that you're with us on this project and get some kind of feedback at the end. You can end it right there. For those of you that would like to continue working on this project for other purposes, you're welcome to do that for however long you have with the license that you have with the program. Any other questions?
Ilya, if I may add something.
Yeah, go ahead.
Yes, so everybody, you will all have access till August 15. So once this week is over, you'll have till August 15 to complete the courses and also work on your project. So if you have any questions regarding that, you can let us know. And also, like, if you want to ask, like, you know, I don't have access, what do I do if I'm not able to complete a course by August 15th, or I need additional access? We will be talking about it, and I'll be emailing you what other things that you can do, like join a program or how you can purchase a license. But till August 15, you have time. So please utilize that to work on your project and the courses. And another thing that I want to add is this, that maybe some of you may not be able to log into your courses today, yesterday, or tomorrow. There is a technical issue that we are currently facing. So that can bar some of you from accessing some of the resources, but you will be able to get back to them very soon. So we are sorting that out. Okay.
Okay, so the question here, where will the presentations be held? Yeah, these will be virtual presentations. There's not any sessions in person right now. And so the first step to get some feedback from the judges is to be able to upload a recorded video presentation, preferably not just slides, but some explanation of the slides. And as you work on those, we will ask you to do that before the submission so we can do a preliminary review, identify any kind of opportunities to improve. So throughout the next few days, as you start working on your slide deck and your slide deck grows, we'll be giving you opportunities to present in front of our class, answer any questions, get some feedback just internally, and make improvements to the slides overall. Once you make those, you'll be able to submit the final video presentation that is recorded and is sent out emailed to these judges. These are judges that are professionals, and they have their own schedules. We're not going to be able to necessarily bring everybody together just for the presentation to listen to everybody's presentation. So we will send it to them, and once they give us the feedback, we will share that back with you. Any other questions? So they're for the next weekend, before, before Sunday next, or yeah, before next Sunday. How to load video with a Mac? Could you do that for Windows? I don't have Windows myself, but I can arrange either a recorded version to share with you. Okay, yeah, we will share with you some of these examples. We have quite a number of them, so we'll post an update for those of you that want to look at these examples. For those of you, again, some of these examples are in a poster format because they were presented at a conference. Others are presentations, recorded presentations, similar to what you just did, right? Just slides that have a certain structure of an introduction, methods, results, and conclusions, and then they are recorded. So we will share that with you on the forum so that you can just take a look at a few examples. And yeah, so Muiwa right there, Chandrani answered, in PowerPoint, you can just record a slideshow and export it as a movie, as a video. Okay, so for any other kinds of technical questions, please be in touch with us on the forum. We will continue this tomorrow. Thank you for joining today, and we'll see you again then. And in the meantime, if you have any questions, post them on the forum. Thank you.